The tools that astronomers use most often are their eyes and cameras. So that's our topic for today. Light, whether it's emitted from an object or reflected, naturally travels outwards. Now I have a light bulb here. It is emitting light. Light is hitting me and reflecting off of me. But whether it's emitted from an object or reflected off an object, light naturally diverges, it naturally spreads outwards. And light from this light bulb is hitting this piece of paper. And so my question is, how come there is no image of the light bulb on the paper? I don't see a picture of the light bulb on the paper, even though light from the light bulb is clearly hitting the paper. In the animated diagram that I have down below, these circles spreading outwards are the wave fronts. Those are the light waves traveling outwards. And I've also drawn 4 arrows up, down, left, right, just to indicate rays. But of course, those arrows, those light rays are going in all directions. Remember, the rays are perpendicular to the wave fronts. And the line there on the right is meant to represent the paper. So the reason there's no image on the paper is that one spot on the light bulb, the light coming from that one spot, is hitting everywhere on the paper as the animation is showing. And light from a different spot on the light bulb is hitting also everywhere on the paper. And therefore there is no image. So to create an image, we need to have light rays from one point on the object go to one point on the screen, and light from a different spot on the object to go to a different spot on the screen. So we need some kind of a device that will cause diverging rays to instead converge or come together. And there are 2 things that will do that: convex lenses and concave mirrors. Here's a concave mirror. They're bent and light rays coming in. Now the light rays coming in are shown to be parallel to each other, which is what the light rays would look like if they were coming from an object far away. So the light rays come in parallel, and then that concave mirror causes the rays to come together. The distance from the center of the mirror to where those initially parallel rays cross is called the focal length. And a convex lens accomplishes the same goal. It causes rays that are, in this case, parallel, but they could even be diverging. Rays that are parallel causes them to come together to converge. And again, the distance from the center of the lens to where those initially parallel rays cross, that's defined as the focal length. Now, to record the resulting image, the image has to be directed onto a detector. The classic detector, the old-fashioned detector, was photographic film or photographic plates. The plates, photographic plates, if you've heard that term before, were actually plates of glass that the light-sensitive emulsion was painted on. And but since the 1980s, astronomers have been using electronic chips or detectors. Of course, those became common in consumer cameras after about the year 2000 or so, a couple years later. So if you have a camera now, I'm sure it's almost certainly an electronic camera rather than a film camera. Now the detector has to be protected from extraneous light. The only light you want to get to the detector is the light that went through the lens. And so you need to put the detector in some kind of a box. And a box is another way of saying a room. And the Latin word for room is camera. And it actually comes from the phrase camera obscura, dark room. And but people are lazy and people shorten that to camera. And that's the origin of the word camera. Now the human eye is in essence kind of a camera. Now it can't record or save the image. So in a sense, it's kind of like a video camera that's just going to a TV screen and but not being recorded anywhere, just displayed on the screen and lost as soon as it's displayed. But this diagram, feel free to pause the video to read the text. I have a diagram here of the eye, so I'm just going to go real quickly over it. This is our last slide. And we have a lens here. And one point I like to make about how the eye works is that the lens of the eye does not actually do most of the converging of the light. Most of the bending of the light actually happens at the air to cornea interface. The cornea is the very front window of your eye. If you wear contact lenses, your contact lenses are sitting on the cornea, in contact with the cornea. And so most of the bending of the light actually happens when light enters the cornea. What the lens does is it fine-tunes the focus. As objects move farther from you or closer to you, you have to adjust the focus. And that is what the lens does. The lens can change its shape. There are muscles on the side and little ligaments attaching the muscles to the lens that allow the lens's shape to be stretched or allowed to fatten or stretch. And that's something that camera lenses don't do. Camera lenses, generally, made out of plastic or glass, are not flexible. And if you have a camera, the way a camera that focuses is it physically moves the lens closer or further from the film or the electronic detector. It changes that distance. But the eye doesn't work that. The lens does not bulge out, move away or towards the retina, but rather it changes its shape. The pupil is a hole in a circular muscle called the iris, and that controls how much light enters the eye. If it's very bright, you can get away with a small hole. In fact, you want a small hole so that you don't overpower the retina with too much light. But if it's dark out, you want to make that hole big to let in as much light as possible. So I've mentioned the retina a couple times. The retina is the detector for the eye. It's the back surface, inside back surface of the eyeball that is lined with cells that are light sensitive. Cone cells and rod cells, this refers to their shape. Cone cells actually come in 3 different varieties that are sensitive to the long end of the visible spectrum, like red, and but also orange and yellow. And the middle part of the spectrum, we often say green, but really the whole middle part of the visible spectrum. And then the short wavelength end of the visible spectrum, the violet, the blue end. So we have 3 kinds of cone cells that allows us to perceive colors, and then there are also cells that are rod-shaped that respond to all the colors of light, allowing us. They're more light sensitive. But they don't distinguish colors. But you may have noticed that in twilight or at night in low light inside a darkened room, you really can't see color very well, and that's because in those low light conditions, you're basically only using your rod cells, not the cone cells. There is an optic nerve that all the data from those cone and rod cells, which are light sensitive nerve cells, those nerves get bundled together and head to the brain. Right where the optic nerve leaves the retina, there's actually a little patch where it is blind. You have a blind spot on your retina right where the optic nerve leaves, because there are no cone or rod cells right there. It's just too crowded. The fovea is the central part of the retina. That's where you have the sharpest vision. The cones and rods are packed most closely together, and in fact, that's where the cones are really concentrated and have far fewer rods. On the outer parts of the retina, you have more rods than cones. And the last thing here I'll just mention is the eyeball has to maintain its spherical shape. It can't just be a deflated balloon. And so there is a clear gel called the vitreous gel that fills the bulk of the interior of the eye. It's transparent, and but it provides the pressure that just keeps the eyeball in its spherical shape. So that's an overview of images, cameras, and the human eye, essential for understanding these tools that astronomers use.